There we go. All right, we are recording now. So here we go with, going to finish up the Cnidarians, talk about a few other phyla before we're done for the day. So here we've got, there's that. Here we've got the material I want to cover, and I need to apologize for a slight goof up in that on Monday we started talking about Cnidaria, and I went over Anthozoa, Scyphozoa, and Cubozoa, but for some reason I left out including the slides that I have on the basic nature of what Cnidaria are, and I didn't realize that until I was about halfway through. So I've already introduced some concepts of what Cnidaria are, but I didn't finish the story, and I've got some general material that I need to go back over before I can finish off the phylum. So we talked about Cnidaria as having a gut with only one entrance, the way out's the way in. A sac-like gut, although the sac may end up being branched, sometimes forming a network of canals and sometimes divided by these partitions, the septa. And I mentioned the biphasic life cycle where many Cnidarians, not the Anthozoa, but all the others, alternate between a sexual generation called a medusa and an asexual generation called a polyp. And the medusa is what you think of as a jellyfish. So we did cover that. What I neglected to mention was that Cnidaria are said to have a tissue grade of complexity, which just means that there are true tissues, meaning that you have cells that are organized into sheets that are held together by gap junctions, tight junctions, and a basement lamina, a kind of felt-like layer of mostly collagen type 4. So there's tissues there, but Cnidarians aren't considered to truly have organs. You don't have multiple types of tissue integrated into a functional unit. You know, your heart, for example, has nerve, muscle, epithelium, and connective tissue, you know, all integrated together into this thing that does a specific function, you know, to go lub-dub, and pump your blood and all that. Cnidaria don't really have anything comparable to that. They're usually considered to lack true organs, although they have specialized tissues that do various things. What coordinates the body is a nerve net, but there's no central nerve center. There's no brain. Nerve cells conduct impulses equally well in both directions. As you'll see tomorrow, if everything goes well, if you take a sea anemone and you poke it, the entire body contracts. Excuse me. Impulses spread across the entire body in all directions from the point where you poke an anemone, and there's no central processing center, you know, that sorts everything out and limits the response to only a part of the Cnidarian body. Everything goes through this network, and the nerve cells conduct in both directions, unlike yours. There aren't true muscle cells, but there are epithelial cells that have these branches that have contractile filaments. So this is myoepithelium, epithelial cells that have parts that kind of do double duty. And the symmetry is primarily radial. I'll talk about that in just a jiff. Almost all Cnidarians are marine. One that it's possible that we'll see in here, although I don't see it regularly enough to be sure of telling you where to go look, is a freshwater polyp in the genus Hydra. I'll show you Hydra a little bit later in this presentation. There are even freshwater jellyfish that pop up in lakes all over the eastern United States Spotting them tends to be just a bit chancy. Some years you find them and some years you don't. And even if they're present, they're transparent and not that easy to see. So many people have never seen them. We do have them in Arkansas, but again, we don't have them consistently enough that I could tell you, hey, go to Lake Beaver Fork on a nice sunny day in August, and you're bound to spot freshwater jellyfish. You can't be sure of seeing them at any given time. Hello, Fern. Right. Now, I mentioned radial symmetry. Radial symmetry, like this octocoral polyp, just says that the animal's got a top and a bottom, a mouth end and an end away from the mouth. Here, the mouth end or the oral end is facing you, and the aboral end, the end away from the mouth, is pointing away from you. But there is no fixed left or right. And you could take a certain number of planes that would pass through the central axis that would divide the animal into equal mirror image halves. So if you were standing in front of me and I were to completely lose my mind and grab a samurai sword and swing it right down on your head going bonsai or something like that, I would never really do this, but in theory, I could slice you into two halves that are of equal size and mirror images of each other, or close enough to mirror images that it makes no difference. Again, I would not do that, but I could, in theory, if you cheated on my test. And there's only one way I could do that. There's only one plane in which I could divide your body to get equal mirror image halves. With a critter that's got radial symmetry, there is more than one way that I could do that. You could take a scalpel and slice this polyp through multiple planes that all would pass through the center, and they'd all divide the polyp into mirror image halves. That's radial symmetry. Let me see. I just saw something go beep, so I'll try to get the cat's behind out of my face and see what that is. One moment. Okay, Garrett joins us here. Good morning. We were just talking about cnidarian features in general, and I was talking about radial symmetry. And we can talk about radial symmetry as coming in varying degrees. There's a moon jellyfish seen from below on the left. That's a pretty common and pretty typical scyphozoan. You can see those four things that look like clover leaves. That's the gonads. They don't normally shine. That's just a trick of the light. Arranged around a mouth in the center, and you can actually see the mouth here. It kind of looks like an X. And we say that Aurelia has tetraradial symmetry because there's four angles that we could rotate that picture and get the same picture. We could take that picture and turn it 90 degrees or 180 or 270 or 360, and we'd get fundamentally the same picture. Okay, slightly different because the bell is flexible and it doesn't look perfectly round, but close enough for what we need to do here. So that's tetraradial symmetry. The solitary coral on the right has hexaradial symmetry because you could do the same thing if you turned that through 60, 120, 180, 240, 300, and 360 degrees. Now, this one's actually not perfectly hexaradially symmetrical because you might notice it's just a little longer along one axis than the other. So we'd actually call this biradial symmetry, which means close to radial symmetry, but stretched or compressed just a little bit in one direction. And that's pretty common among Cnidaria. But this is pretty close to what hexaradial symmetry would look like. There's 6 angles you could turn that drawing through to get a drawing that looks the same as the one that you're starting with. Okay, there's two basic body forms. Yeah, I think that's material I've already gone over. Tissues have this basal lamina of collagen, two layers of cells with mesoglia in between, like a grape jelly sandwich. There's stings. I'll talk about those stings in a bit, and the sac-like gut with a mouth but no separate anus. The two basic body forms are on the left. You've got a polyp, which has the mouth facing up, the oral end down, and tentacles surrounding an oral disc. And then the medusa is a lot like the polyp, except the mouth typically faces down, and the tentacles hang down, and medusas swim. Fundamentally, they're very much the same structure. They've got a mouth opening into an open sac-like gut with a mouth surrounded by tentacles. The difference is that the medusa is generally sexual and the polyps generally asexual if you have that biphasic life cycle. So you saw a picture of it on Monday. There's a critter called an upside-down jellyfish, genus Cassiopeia, that actually sits on the bottom of shallow, muddy or sandy beds in places like coastal Florida and the Caribbean. And it's a medusa that thinks it's a polyp. We still call it a medusa and not a polyp, first of all, because it's the sexual generation, and second, because it's still capable of swimming if you disturb one, even though it doesn't do it very well or very much. So we still classify it as a medusa on the basis of structure, position in the life cycle, and capability, even though it's a medusa that behaves a lot like a polyp does and that it spends most of its time just kind of sitting there. This is an Antarctic jellyfish that I picked because you can really see the two tissue layers. The gastrodermis is red. So the interior of this jellyfish, you can see it's a reddish-orange color. There is a mouth in the middle of it, although you can't see it very clearly here. And then you're seeing a bunch of canals radiating out from the stomach, which is kind of up at the top of the bell. All that red stuff is gastrodermis. And then surrounding it, you can see a transparent layer. That's the epidermis plus the mesoglia, which is in between the epidermis and gastrodermis. So this particular picture just shows you those two tissue layers very well, I think. And then there's a slice through the body wall where you've got epidermis, mesoglia, and gastrodermis. The gastrodermis secretes enzymes and things like that. Both tissue layers have epithelial cells that have these extensions that kind of come down and form muscle-like contractile fibers. That's what I mean by myoepithelium, where the cell body is epithelial, but then this extension of the cell down below is packed with the contractile fibers. And then there is a cnidocyte, a type called a nematocyte, kind of in the foreground, and let me talk about those. The sting of a jellyfish or other cnidarian is called a cnida, and they're intracellular structures, and they're very complicated secretions. They're technically not organelles, although they've got rather complicated structure of their own. They can range from 5 microns to 100. They're secreted by the Golgi bodies of specialized cells called cnidoblasts. And once a cnidoblast has secreted a cnida, it's called a cnidocyte. And if anybody forgot, cnidos is Greek for a stinging nettle, a plant that's got stinging hairs. All cnidarians produce a specialized type of cnida called a nematocyst. And nematocyst, nema is Greek for hair, and nematocysts, when they pop open, they eject a very long barbed hair that injects typically a neurotoxin into whatever has been trapped. So this long hair not only penetrates prey and sticks to it and traps it, it also injects venom. All Cnidaria, there might be a few goofy exceptions, but virtually all Cnidarians have these. Most of them can't actually penetrate human skin. You could handle sea anemones, for example, all day long, and it wouldn't bother you because those Cnidae simply don't sting hard enough to penetrate your skin or inject any toxin that would do you any damage. There are, of course, some jellyfish that can do you damage. There's some that can even kill you. So exactly how dangerous these critters are depends on the species you're talking about. Get your tail away from the camera, Fern. Nobody wants to see that. Good kitty. There are a couple of Cnidarians that have other types of Cnidae that they use for other functions. One order has what are called spirocysts, which don't wrap around, they don't inject toxin, they just tangle up prey and wrap around them. And one group called the tube anemones has Cnidae called tychocysts that eject these sticky threads that they use for building the tubes that they live in. They don't use them for prey capture. But all Cnidarians have nematocysts, which are the type of Cnidae that are specialized for stinging and paralyzing and killing prey. Over at the left, there's an unfired nematocyst shaped like a long oval, and you can see there's kind of a tangled thing in the middle. This has been removed from the cell that made it, by the way. They're pretty easy to pull right out of the cell. And you can see it's, round. And there's a trapdoor on one end. And on the right, you can see a nematocyst that hasn't fired. And you can see that filament is coiled up inside. The one on top has not fired. The one on the bottom has. And what you can see is there's this long hair-like thing that is shot part way out of it and is continuing to uncoil and shoot its way out. That's a nematocyst that has fired with that trapdoor popping open. Right. This is a diagram of one in the process of firing. There's a trapdoor on it called an operculum. You'll see that word again for a trapdoor-like structure that swings open very rapidly, and a piercing part of that filament bursts its way out extremely rapidly. Takes less than a microsecond to do it, and that typically will penetrate the skin of whatever is unfortunate enough to get stung. And this is kind of hard to show in the diagram, but that coiled up filament actually begins to come out the center of that piercing, penetrating part and turn itself inside out extremely rapidly. Kind of like turning a sock inside out, except the sock is extremely long, and the turning inside out process is extremely rapid. It may take less than a microsecond. But the end result is that the prey is stuck. It's got this filament that may actually penetrate into the body, and the penetrator and the filament have injected toxin into the prey. So if you're a typical Cnidarian and a little shrimp brushes up against one of your tentacles, the shrimp is not only snagged, it's going to be paralyzed and stop moving very quickly, and then you can feed on it. And there's one that's already fired, and you can see that you have this barbed little filament sticking out. Now, Cnidae are energetically expensive to make. They are complicated structures. It takes a lot of energy for a cell to make them. And it's not very adaptive for, let's say, a sea anemone to fire off a whole bunch of nematocysts when it's just brushed up against a rock, right? You know, there's no point for a jellyfish to try to sting pebbles to death. So there are sense organs that prohibit a cnidarian from firing until it's until there's something that is actually likely to be able to eat. So on each cnidocyte, there's typically a modified cilium called a cnidocyl that acts as a trigger. The cnidocyl bears mechanoreceptors that mean it's likeliest to fire when it's vibrated at a particular frequency. There's one particular frequency that it can pick up, and that usually tends to match, oddly enough, the swimming stroke frequency of the local shrimp. So if a typical shrimp is beating its appendages at 30 times a second, the local cnidarians will often be tuned to fire their stings if they're vibrated 30 times a second. The cnidocytes also have chemoreceptors that will trigger firing in the presence of chemical signals as well. So an anemone, let's say, will typically not fire if you drop a pebble on it, but it's much more likely to fire if you drop something that is made of chitin or of protein, because that's what's typically present in the skin of the little shrimp or fish or whatever that it's trying to feed on. And this is cool because that's a sea anemone tentacle. And these are what the sensory receptors look like. You have a central cilium called a cnidocyl. That's the trigger. And the mechanoreceptors are present on these microvilli that surround the central cilium. And this ought to be looking hauntingly familiar because it's the same arrangement as you get in sponge choanocyte cells. And it turns out that that arrangement of a single cilium surrounded by microvilli is not only an effective filter feeding mechanism, it turns out to be a fairly effective way to make vibration sensors. Because if that central cilium vibrates back and forth, it can touch the surrounding microvilli and set off an electrical potential. So the mechanoreceptors of sea anemones seem to be an inheritance from choanoflagellate ancestors, the same basic ancestry, the same basic structure. And the really cool thing is this. There's a guy at University of Louisiana in my hometown of Lafayette by the name of Dr. Glenn Watson, who's found that these mechanoreceptors on sea anemone tentacles look exactly the same as the hair cells in the mammalian cochlea, the part of your inner ear that actually detects sound vibrations and passes nerve signals onto your brain. Those also consist of a single cilium surrounded by a ring of microvilli. They look exactly like this, and they function in basically the same way, and they're both used to detect vibrations. Yours are a bit more sophisticated than the anemones are. And what he's been doing is using anemones as model systems to look for ways to treat hearing loss. The cells inside your cochlea are very hard to reach because they're buried deep in the petrosal part of your temporal bone. They're buried in this really thick part of your skull that you can feel just back of your own ear. They're deep down in there. You can't reach them without drilling. People tend not to like it when you drill through their skulls to get their cells for study. And it's also very hard to keep them alive outside of the body for any length of time. So it is not easy to work with mammalian hair cells. They're hard to get to and die pretty fast when removed. So you're limited in the experiments you can do, certainly on humans, but also on mice or what have you. Anemones you can easily grow in the lab, and you can get, right there. The tentacles are just right there hanging out on the outside. There's nothing you have to cut through to get them. And if you feel bad about cutting anemone tentacles off, it's okay because they regenerate. And so Dr. Watson and his colleagues have been studying anemone receptors as a model system to look at hearing loss. And last I looked, a couple of years ago, that actually found some proteins that anemones use to repair their own mechanoreceptors, which at least in lab experiments so far look like they might have the same function in vertebrates. So this is being looked at very seriously as a treatment for certain types of hearing loss. In particular, the kind of hearing loss that you get when you're exposed to extremely loud sound. The kind of thing that you might get if you've been exposed to an explosion, or, played guitar in front of a giant rack of Marshall amplifiers every night in front of screaming crowds for 20 years. This might actually fix, what poor old Pete Townsend is dealing with. So certainly potentially quite beneficial, and yet another good reason why you pre-meds ought to know something about invertebrates. Every so often you can find model systems in perhaps very unlikely places that have, very direct impact on human health and well-being. So there. Now, the last class I'm going to talk about is the hydrozoa, but I'm actually going to pause recording right now. I would keep going. I've got 9:41, so I'm going to come back at 9:50. If you've got questions or anything like that, put them on the chat. So I'm going to pause the recording now.